Hello everyone, my name is Molly. I'm a 5th year grad student in the Bautch Lab, and I'm here to take you on a tour of our lab today. So our lab is led by Dr. Victoria Bautch, and we're rather a big lab. We're made up of a bunch of grad students, postdocs, and undergrads who all work really hard but help each other out, and so it's a great group of people. Here in the Bautch Lab, we study angiogenesis, which is the process by which new blood vessels are formed. First things first, I'll take you through the main lab. So when you first walk in, on the left we have shelves full of glassware. This is graduated cylinders and bottles, everything we need for holding and measuring liquids. Next up, we have the first of many freezers. This one is at -20 degrees Celsius, which is just like the freezer you have at home. So in it, each of us has a rack with a bunch of boxes full of samples. This box is holding my antibodies, and you can see I have everything nicely labeled so it's easy to find when I need it. Over across from the freezer, we have a sink with the eye wash station. Then next to that, we have a water bath that we usually hold at 55 degrees Celsius, and then a heating block where we can get things up to boiling. After that, we have a microwave, and then a magnetic stir plate that doubles as a heating plate, and a vortexer for mixing up samples. Next to that, we have a desktop centrifuge, which spins down little strips of tubes. This is next to the PCR bench, where we do all the PCR for our lab that tells us which mutations our mice have. Across from the PCR bench are the machines where the PCR is actually done. So these machines change the temperatures and allow amplification of DNA. So since this one is done, we can see what's in the machine. So this is a strip of PCR tubes. The DNA is inside there, and we have a green dye which helps us see it when we run it out on a gel. Beyond the PCR machines are benches where people sit and do work. And that is an inflatable dinosaur that came as a prize with some of our supplies that we ordered. Scientists have fun too! Here's a typical example of a lab bench in our lab, complete with everything a scientist needs to do research. We start off, we have a lab notebook on the left. This is where we keep detailed notes of our experiments. Up here we have pipettes, and next to them the pipette tips, and these help us measure small and precise amounts of liquid. To the right, we have a vortex, which mixes samples, a centrifuge, which spins them down, and over here, I have samples on a rocker. Let's take a peek at what's under the foil on this rocker. It happens to be 2 samples of livers, which is a great organ to study for angiogenesis because it has so many blood vessels in it. Now I'm going to show you how we get the samples of liver, and then what we do afterwards to study and image it. This is a dissecting microscope. It provides kind of low magnification and a good light source, which is good for when you're dissecting small tissues. And we take our samples and we put them in a petri dish like this one. We fill the petri dish with PBS, or phosphobuffered saline. And this is just a saltwater solution that keeps the tissues from drying out while we're working with them. So, like I said, we fill up the petri dish with our PBS, and we put our sample in there, and then we dissect it with tools. Here are the tools we use for dissection. On the left, we have fine forceps. These allow us to do tiny manipulations and hold the tissue. Next, we have a pair of small dissecting scissors. And on the right, we have a pair of spring scissors, which allow for finer manipulations if you have to cut something very tiny. This is Lauren. She's a postbac in the lab, and this is her dissecting something right now. She looks through the dissecting scope to magnify what it is because everything that we dissect is too small to see with your eyes. Once we've dissected the tissues out, we fix them and then send them to a core on campus that embeds them for us in these blocks. So here's a cassette with a sample of liver in the middle, and it's embedded in paraffin wax, which is just like candle wax. So what we want to do next is to slice these samples very, very thinly, And to do this, we use a piece of equipment called a microtome. You can think of it like a deli slicer. So we put our sample in the block up here, and then it takes a very, very sharp razor blade. And as you turn the handle to the right, it advances the block forward 10 micrometers at a time, so very, very thinly. And then it makes these slices that we can put on slides. Here's a box of slides with the tissues on them. The tissues come out in a ribbon from the microtome, and so we can fit several samples on one slide. This allows us to look through the sample in the middle of it and see details that we wouldn't be able to see just by looking at it from the outside. This is great for studying blood vessels because blood vessels are all throughout the tissue. The tissues come out on the slides clear, which isn't very helpful for seeing what's going on. So one of the common stains that scientists use to see the tissue is called an H&E, or hematoxylin and eosin. So these are two chemicals that will stain the tissue various shades of pink and purple and help you see what's going on. So you load up the slides in these cradles and you dunk them in the various solutions of chemicals. At the end of this stain, you're left with very pretty pink and purple sections, like these ones here. After the stain is done, we take the slide to the microscope so we can see what's going on at a small level. There are different objectives on this microscope, and each one of them magnifies a different amount. We have 10x in yellow, and that magnifies the thing in the view 10 times as much as it would be with your eye. We also have 20x and 40x for seeing very small details. Once the slide is on the microscope and you've chosen the objective you want to look at it and focused it, you can either look at it through these eyepieces, or this microscope happens to have a camera built into the top of it with a view screen that lets us see what's on the microscope right now. So this is what our section of stained liver looks like. You can see the blood cells are this lighter red color here, and they are contained within a blood vessel, which is this circular structure surrounding the blood. You can also see a bunch more blood kind of dispersed throughout the rest of the liver, and that's pretty characteristic of a liver at this stage in time. I've already shown you two of our microscopes in lab, first the dissecting microscope and then the upright microscope where we're looking at slides, but we have several others that we use all the time. When you walk into the microscope room, on your left we have another dissecting scope. This is similar to the one that we saw already, but this one not only has a camera attached to the top where we can take pictures of what we see, but it also can let us see fluorescent things. So that's what this box does. It's the laser source for the fluorescent scope. On the other side here, we have what's called an inverted microscope. So before, if you remember the scope with the slides, it had the objectives coming from the top. This one has them coming from the bottom, underneath the stage, which lets you look at thicker samples, so for example, cells in a big dish. The last scope in this room is our big expensive one. This is called a confocal microscope, and it takes really crisp, clear pictures of very small structures. All of these boxes to the left are part of this scope. Part of their function is to emit laser light at different wavelengths. This allows us to do antibody staining with up to 4 different antibodies and see them in effectively 4 different colors. So with that, I'll wrap up our tour today. I hope you enjoyed getting to see the space where all of our research is done, and happy to take any questions.